Thank you so much, Dan and Claire and Russell and Sarah, for inviting me. It's actually a huge honor to give the first lecture of the Gatsby Plant Science Summer School this year. And I mean, it's great because I can feel the energy to be surrounded by all of you. Welcome to the Gatsby Plant Science family. I was a member, as Dan mentioned, a very long time ago. And actually, I remember when I was in your shoes, not in the Gatsby Plant Science Summer School, but when I started becoming interested in plant biology as an undergraduate student. So today I'm going to share some of our research interests in my lab, and also I'll touch upon what inspired me to be interested in plant biology, and in particular plant photobiology. So as you can see here, that's just a photo I took last week when we went for a trail, the Gruffalo Trail with my son and my husband. And usually I don't show photos of nature, but actually it's very important, especially the research that we do in plant biology, to realize that plants, of course, unlike animals, they cannot escape when the conditions are unfavorable. And so they have to be able to adapt and respond in order to survive. And in particular, light and temperature are environmental parameters that are constantly fluctuating. So this is something that we work on, and I'm going to give you an introduction on light sensing in plants. I'm sure many of you are familiar with plant photoreceptors, many of you maybe not so much. Then my journey in plant photobiology, and then I'm going to talk about a particular protein that we're interested. We call it tandem zinc finger plus 3, not very imaginative, but we call it TZP, and how we believe it integrates signals, light and temperature, in order to regulate two very quantitative responses in plants, in particular in Arabidopsis, that we use as a model system. One of them is hypocotyl elongation. So the hypocotyl is the embryonic stem. So we study how it grows, and again, this is very sensitive and quantitative assay. And then flowering initiation. So basically when they transition from vegetative to reproductive growth. So again, I'm pretty sure everyone is aware of the fact that light is essential for energy production for plants, that they have to make their own food. But also light is a very important informational signal that regulates almost every aspect of plant growth, and also some irreversible developmental transitions. So light is essential for seed germination, and also is very important for this developmental transition from dark-grown plants to light-grown plants. So when plants emerge from soil, they have very elongated hypocotyls in search of the light, and then as soon as they're exposed to light, there is inhibition of hypocotyl elongation. There's opening of the cotyledons. Again, these are the embryonic leaves, and then there's greening of these cotyledons because they start photosynthesizing. And again, this is irreversible, and over 70% of the transcriptome is basically changed in response to this light exposure. Then light is also very important for synthesizing natural sunscreen. So unlike humans, the plants don't need to apply sunscreen to protect themselves against ultraviolet radiation. So UV light actually induces the production of flavonoids that protect the plants from UVB. And then, of course, light can regulate different responses. So there's stomatal opening and closing depending on the light conditions, the light quality, intensity, direction and duration. And also again, like ourselves and animals and other organisms, plants have a clock, a circadian clock. And light is very important for informing the clock of how much light there is within a 24-hour period. And in addition, another irreversible developmental transition is as I mentioned earlier, is flowering initiation. So again, light and photoperiods, so the length of day within a 24-hour period is very important for determining whether a plant will flower or not. And last but not least, shade avoidance. Again, you may have heard of this response. Basically, when you walk in a forest. You can see that there's plants that are very tall, the trees, and then there's shorter plants under the canopy that, again, they are in search of light. So plants need to be able to determine whether they're growing under a canopy. And it's not just the fact that there's lower light intensity, it's actually the quality that changes a lot. So the red to far red light ratio is different because the plants that are at the top, they're photosynthesizing, so they're absorbing all the red light. And this is what the plants under the canopy sense, and this is a signal for them to start growing towards search of the light. But also whether sometimes there's, not necessarily under a canopy, they can sense whether there is another plant that is basically competing for light, that is doing better than them. And again, they need to extend, they change architecture in search of light in order to adapt and then survive. So unlike the rhodopsin visual system that we have and other animals also have, the plants don't only have one protein or one molecule that senses all wavelengths of light. They need to sense not just light wavelength, so light quality, but also duration, direction. So they have evolved 5 different families of photoreceptors. Each family basically absorbs and perceives different wavelengths of light. So there's one UVB receptor. It's actually the only genetically encoded UVB receptor in nature, and it's found in plants as UVR8. So it senses UVA, UVB. And then there's 3 families of blue light receptors, the cryptochromes, the phototropins, and the Zeitlupe family. And phytochromes, there's 5 of them in Arabidopsis, and they sense red and far red light, and also the ratio between red and far red light. So all of these proteins contain domains that can attach to a chromophore, and these basically act as the eyes of the protein, as we call them. So this is where light sensing, light absorption happens, and then there is signaling cascades within the protein itself, so conformational changes, and also downstream signaling cascades that basically relay the signal that initiate changes in gene expression, and then architectural changes and also developmental changes. UVR8 is the only receptor that doesn't have a chromophore, but it has tryptophans that can absorb UV, and that's how it actually acts as a photoreceptor. So all of these receptors have unique but also synergistic functions, and they work together in order to change architecture and development in response to different light conditions. And with the exception of phototropins that are plasma membrane associated proteins, all the other photoreceptors are either constitutively found in the nucleus or move. They translocate from the cytosol into the nucleus upon light exposure. And I'm just pointing this out because, again, in response to light, there's major transcriptional changes and epigenetic changes that are very important for allowing these changes in architecture and development to occur in plants. Now, obviously, sun is the major source of light, and sun is very important for changing the temperature. And I'm sure you will agree that you can see it yourself. There's changes in temperature in the last few years. I don't know about you, but in Scotland, it's the warmest summer we've ever had in 10 years. I come from Greece, and I've never had to walk with a sleeveless top in Glasgow, and this year it's very prolonged. Anyway, I'm not complaining, but obviously, if this continues, it will be a major problem for agriculture, for everyone, and for many different reasons. So in terms of plants and crop production, it's predicted that as little as 1 degree centigrade increase will lead to major crop losses. Of course, this depends on the species, but clearly it's something that we need to look into as scientists and try to help understand how plants respond to these changes in light and temperature, and try to optimize crop loss if we can. And in terms of the different responses to temperature, it's not just at high temperature. Obviously, temperature, depending whether it's low, ambient, or high, it affects different developmental processes. So low temperature is important for seed germination or stratification, and then also prolonged cold exposure is very important for some plant species for vernalization, which is basically the initiation of flowering. Then ambient temperature between, let's say, 18 and 32 degrees is considered as a response that's collectively called thermomorphogenesis. And then at higher temperature for 35 and above, there's heat stress responses. So this is where the plant is basically fighting to survive. But with the increase in temperature, all these basically will shift higher and all these responses will be affected. Now, in relatively recent years, in the last, I guess, 10 years, it's been more and more obvious that there's a connection between light and warm temperature sensing. So this is the thermomorphogenesis temperature range I'm talking about, from 18, let's say, to 32. So a lot of scientists have shown that some of the de facto well-established light signaling components also act as thermomorphogenesis components. So some of the photoreceptors, for example, phytochrome B, has also been shown to be a thermosensor. Also ELF3, early flowering 3, which is involved both in light signaling and clock responses, has also been shown to act as a thermosensor. And these are just some of the light signaling components that have been shown to act in thermomorphogenesis. So it's almost as if the plants have evolved common mechanisms. So instead of using different signaling components, different proteins, it's almost as if they've hijacked this pathway. But I think it's just us discovering that warm temperature and light signaling, they're highly linked. And it took us a very long time to figure this out, clearly. So, and this is an example of some of the responses that are very similar to shade avoidance and in response to warm temperature, so thermomorphogenesis. So in response to shade avoidance, you have extension of this hypocotyl, and also you have extension of the petioles, and also you have this hyponastic shape in the leaves, and you have acceleration of flowering. And exactly the same responses happen when plants are growing in higher temperature than they're used to. So in Arabidopsis, what we call normal temperature or favorable temperature is 22. So when you move them to 27, you will see these responses that are almost identical to shade. So it makes total sense to use the same signaling pathway. The same genes, basically, that are upregulated in response to shade are also upregulated in response to warm temperature. And then they promote the same architectural changes. So basically, the plants have to adapt or grow in terms of depending on the conditions that they are at. And these are players that determine whether one or the other will happen. So in terms of thermomorphogenesis, these changes in architecture basically help the plant dissipate heat, whereas for shade avoidance, it's just searching for light, basically. Now, how I became interested in photobiology, I come from a small island in Greece, because usually we avoid the sun because it's too strong. And at an early age, I was really interested in biology. And I moved to the University of Glasgow. I always wanted to study abroad. And at the time, Glasgow was offering different degrees in biology. And I was really interested in biochemistry, molecular biology, but also marine biology. My dream was to study echolocation of orcas on Vancouver Island. Anyway, it all changed, but I mean, it was actually, we had really good lecturers, first and second year biology, that showed the power of genetics. So we looked at mutants in Arabidopsis that constitutively had photomorphogenic phenotypes in the dark. And that was really powerful to see how a plant growing in the dark can actually look like a light-grown plant, but without the green leaves. And then, of course, how changes in gene expression regulate a lot of developmental patterns. It was something that, I don't know if it was just the lectures, but also the topic, it really attracted me. And yeah, of course, in Glasgow, I appreciated the importance of sunlight and also warmth. But it was actually, I was very lucky because there was a departmental seminar at some point of the late Professor Winslow Briggs, and he talked about these new photoreceptors that they discovered in his lab, the phototropins, blue light receptors, and he talked about how these domains that were attached to a chromophore were actually capturing light. He was showing the photochemistry, so biophysics, hardcore biophysics, and coupled to biochemistry, like in terms of kinase acids, how the signal was relayed to changing the function of the protein, how it was transmitting the signal internally, and then how this signal was leading to hypocortical elongation and bending in terms of phototropism. So for me, the fact that you could use all these different techniques and different approaches to study plants was fantastic. It wasn't just physiology or taxonomy or genetics, it was a combination of techniques. And I really wanted to follow this route. And it was thanks to the Gatsby that I was able to go to Winsel's lab for a year. So he said, yeah, I have some money, but it won't be enough to live at Stanford because it's very expensive. So there was an email from the Gatsby Mentor at the time at Glasgow, and I applied for the Sainsbury undergraduate studentship, and I went to his lab. So basically we were studying the proteins that are responsible for this phototropic growth towards blue light, and we had made each of these lab domains blind, and we were characterizing in terms of physiology and also biochemistry how the protein was functioning and whether you could rescue the phenotype of the mutant that couldn't bend towards blue light. And I had a great time. I met really amazing scientists, and I was very lucky to be in such an environment. And after that, I was determined that I wanted to stay in research and I wanted to do a PhD. And I really liked light signaling. I really liked the community and the fact that you could work also on different proteins on different light wavelengths. So I returned to Glasgow for my final year of my degree. And then when I was looking for PhDs, there was a project in the lab of Professor Gareth Jenkins at Glasgow. Again, it was on a protein that at the time wasn't shown to be a photoreceptor, and nobody believes that there is a UVB receptor in nature. So my project was to characterize this protein at the subcellular level, and basically the mutant plants would die if you expose them to UVB, whereas the wild-type plants wouldn't. And my project was, yes, to see how this protein worked. And again, I really enjoyed it. Of course, there were difficulties in every PhD project, usually there is, but I wanted to stay in photobiology. And I was very lucky. Again, I was a Gatsby-supported PhD student, and you can probably recognize some people here in the audience. So I think Liz is one of your tutors, and I think you can see the plenary speaker there. So we overlapped during my PhDs. And again, it was a great community to be part of. So it's very important to have a peer network that you can support each other and also exchange experiences, ideas. And, you know, every time we meet, we feel that we are part of the same family. And that's some of us trying to write Gatsby with our bodies. I don't know if you can tell that it says Gatsby here. And I think we are helping, probably that was the second, the Gatsby Plant Science Summer School a long time ago. And I think we had as much fun as the undergraduates. So anyway, so after my PhD, while I was applying for postdoc fellowships, again, there was a Gatsby supported project awarded to Professor John Christie, and that was back to phototropin. So I moved from blue to UV and then again blue, and this time we were purifying these light sensing domains and trying to use them as fluorescent reporters. So usually most cell biologists use GFP as a fluorescent reporter, but there are some disadvantages for specific experiments. So we use the lab domains. They're a third of a GFP, and you could actually label viruses, and you can monitor viral spread. And we were characterizing these domains also, how good they are in terms of fluorescing and whether they bleach. And so there were some advantages over GFP. So again, it was different approaches, different proteins, different light conditions. And eventually when I decided to go to the Corey lab for my postdoc, it was more that I realized that signal integration is as important as working on each photoreceptor in isolation, because in reality, plants are exposed to different wavelengths of light all at the same time, and these photoreceptors need to kind of talk to each other, so there's crosstalk that happens further downstream. And some of the components that integrate these signals are very important for the downstream signaling effects in terms of gene expression. And again, I was very lucky. I was in a community of very ambitious scientists, and I learned a lot from being in that lab. And I worked on a protein that I'm going to talk about today that was originally identified by a brilliant geneticist, Olivier Ludet, who basically used natural variations. So he collected Arabidopsis natural variants from all over the world. And then he did some genetic crosses to identify new loci that were not identified in previous screens in terms of hypocotyl elongation in response to different light conditions. And this particular protein was identified by his screens. So this locus, locus 5 light locus, basically had a premature stop codon, and that led to the shorter hypocotyl phenotype in one of these two in Bay, whereas Shagdara had basically didn't have a mutation and the plants were more elongated. And so that's when I started working on the protein, because Olivier had his own lab and he just wanted to discover new genes, where I was more interested on how the proteins work. So it's good because it worked well for both of us. And again, Joanne was very generous and she allowed me to take the project with me when I started my own lab in Glasgow. So basically we use as a model system Arabidopsis. It's just a weed, but in terms of studying complex biological questions, I think it's still a great model system. And we mostly look into light-induced responses to photomorphogenesis, flowering initiation, and more recently into light and temperature crosstalk. We use different approaches, like many labs in the UK, so transcriptional regulation using chromatin immunoprecipitation assays and RNA-seq analysis. We do some imaging, both confocal imaging, that again, you'll be very lucky that you will be exposed to the confocal microscopes and the lab practicals that have been prepared for you this week. And so this is to study dynamics, so how proteins move in response to light and temperature, and whether they co-localize or interact with other proteins. And of course, some structure function analysis, so basically understanding how different domains of a protein are important for its function. So obviously, having a structure of a protein is very, very important. And now with AlphaFold, we can predict the structure of any protein that we're interested in. However, you can still get a lot of information by just looking at the amino acid sequence of a protein. So in this case, this is DZP that got its name based on its domain composition. And you can see more than half of the protein, you can see in yellow, is predicted to be unstructured. So there's no domain, there's no special beta sheets, alpha helices, nothing. So it's predicted to be disordered. And then towards the C terminus of the protein, in blue, there's 2 zinc finger domains and then a +3 domain. So we know from mass spec analysis, there is a lot of post-translational modifications at the disordered part of the protein. And then the zinc finger domains are highly conserved in nature. They're found usually in transcriptional regulators or transcription factors, and zinc finger domains are usually sites of binding with nucleic acids, so they can associate with RNA, DNA, or other proteins. +3 domains are less well characterized, so they're also highly conserved from yeast to humans, and proteins that contain +3 domains in humans are involved in chromatin remodeling or co-transcriptional splicing. So basically, regulation of gene expression, more or less. So we had an indication that this protein must be involved in regulating gene expression just based on its domain composition. And again, there's specific motifs, like this one is a putative ear motif, that it's a plant-specific motif that usually recruits transcriptional co-repressors, again, suggesting that possibly this is a protein that affects gene expression. And we and others have shown that DZP regulate hypocotyl elongation in response to blue light. So the mutant has less elongated hypocotyls, whereas when you overexpress the protein, you have longer hypocotyls. So it's a positive regulator of hypocotyl elongation, whereas in response to red light, TZP associates with the photoreceptor phytochrome B, and they promote flowering under long day photoperiods. However, in response to far red light, it's been shown that this protein actually acts as a negative regulator of hypocotyl elongation. Again, suggesting that depending on the context, TZP has different functions. In terms of the blue light regulation of hypocotyl elongation, Giorgio, a postdoc in my lab, has shown that TZP can actually associate with promoters. We believe that this is not directly, it's by associating with a canonical transcription factor, zinc finger homeodomain 10, and they bind to light-regulated elements. So promoters tend to have different elements depending on what they're usually regulated by, what kind of stimuli. And CAC GTG is a very common light-regulated element, and that usually is found in genes that promote hypocotyl elongation. Whereas in response to red light and through association with Phi B, we've showed that TZP can again associate with promoters, in this case FT, so flowering locus T is one of the major florigens that induces flowering initiation in Arabidopsis in response to long days. But also recently, Elisa and Giorgio showed, based on ChIP-seq and RNA-seq analysis, that TZP can indirectly also regulate negative regulators of flowering initiation. So in this case, it can affect the expression of histone-modifying enzymes that apply the brakes. Basically, they don't allow FLC to be expressed. So again, suggesting that this protein, depending on the proteins that it interacts with and depending on the pathway it operates, it can integrate different signals. So in this case, it would be light, but also for FLC, it's temperature, cold temperature. So yes, one protein can have different functions, again, depending on whether it's promoting growth or adaptation or developmental transition. So this protein, we noticed at a very early stage that when you tag it with a fluorescent reporter, it can form these punctate structures in the nucleus, also called nuclear bodies. So it's constitutively found in the nucleus, but in response to white light or red light, so white light is primarily red light and a little bit of blue, it can form these nuclear bodies. So they don't form in response to blue light, just red light. And when you have a closer look using super resolution imaging, so you can see that they form these hollow donut-shaped structures, which is not that common. So not every single protein that goes to these nuclear bodies forms these donut-shaped structures. And actually, the person that when we were doing the microscopy, I asked him, have you seen this before? And he said they look very similar to PML bodies. So this person used to work with mammalian cultures, cell cultures, and PML bodies are known to be sites of transcriptional regulation, but also sites of sumoylation. So potentially, this is an area within the nucleus that there is some kind of regulation that we still don't know. I mean, there's different scenarios and hypotheses depending on the protein that you work on. Now, these punctate structures, recently, they receive a lot of attention based on the fact that a lot of them form through liquid-liquid phase separation. And especially within the nucleus, it makes total sense because within the nucleus, there's no membranes. At least nobody has reported that there's membrane associated structures within the nucleus. So if you have compartmentalization, usually it happens through this liquid-liquid phase separation, which is basically what you see when you mix oil and vinegar if you are at a restaurant. So basically, there's two phases, dense and dilute, and this is what separates these two. And these two phases, the dense phase, could be a higher concentration of protein and nucleic acids, and these can be affected by the structure or the composition of these proteins. So usually proteins that form biomolecular condensates through liquid-liquid phase separation, they have these disordered regions. So they tend to stick to each other, let's say. However, this is usually tightly regulated, and it depends on a stimulus, so it could be light, it could be pH, again, it could be concentration, it could be nucleic acids that promote this clustering into these, let's say, nuclear bodies, and there's specific proteins that act as scaffolding components. So TCP seems to have some of the characteristics of proteins that form these bimolecular condensates through liquid-liquid phase separation. So when you do fluorescence recovery after photobleaching, so FRAP studies, so basically you bleach one of these bodies in the nucleus and you see how fast it recovers, you can see that this happens within seconds, suggesting that there's something actively recruiting this protein to this part of the nucleus. Whereas if you do the same experiment in a part of the nucleus that doesn't have a nuclear body, you can see it's much slower. And also, when you use some pharmacological inhibitors that have been associated with disrupting structures that form through liquid-liquid phase separation, you can see that these bodies are affected. Of course, this is a reversible effect. So from the application to actually the imaging, you need to be really quick because the cell can actually recover from this particular drug. So potentially these TZP bodies form through liquid-liquid phase separation, but it's not very surprising because there's two photoreceptors that have been shown to form nuclear bodies through liquid-liquid phase separation, phytochrome B and cryptochrome 2. And I mean, this is something that the nuclear bodies and the whole concept that there is compartmentalization within the nucleus, it's not a novel concept. It was originally described by Ramonica Hall in 1903. And it is a way to compartmentalize the nucleus because, again, apart from the fact that there's lots of DNA that is organized more or less like spaghetti, and there's the chromatin, and there's all these other proteins that are, well, they have to be able to access specific areas within the chromatin in order to regulate it. So there are specific, let's say, compartmentalization of events. So there's possibilities that this is a site of degradation, protein degradation, protein accumulation, regulation of transcription, and also storage, post-translational modifications, or splicing. Or it could be, all of the above. And TCP has been shown to interact and co-localize with a lot of light signaling and also thermoregulatory components. So TZP nuclear body formation depends on the photoreceptor and thermosensor Phi-B, but also it co-localizes with L3, which is also shown to be a thermosensor. And based on mass spec analysis and also direct interaction studies, we know that TZP is part of a large multi-protein complex that again is found in the nucleus. Some of them are photoreceptors and thermosensors, some of them are transcription factors or regulatory proteins, but basically all of them have been shown to be involved both in light and temperature signaling. So I was invited to a thermomorphogenesis meeting because I was collaborating with somebody that works on thermomorphogenesis, and I gave a talk just on light signaling. And everyone was asking, have you checked if TZP is actually involved in thermomorphogenesis? And initially I thought, well, everybody works on temperature. I don't want to be one of these people that jump on, whatever is fashionable, but it makes total sense. So it was a project that it started with an undergraduate student and it evolved. And basically this is just to remind you that phytochrome B, that is a red far-red light receptor, acts also as a thermosensor. So basically high temperature pushes the back reaction and the protein goes back to the inactive state. So basically as if it's never seen red light. And so it promotes the desensitization of phytochrome B, high temperature. In the case of L3, it's a little bit different. L3 is not a photoreceptor, but it's a signaling component. So it acts downstream of the photoreceptors and the clock. And high temperature basically makes this protein form these condensates in the nucleus. And the protein is not as active anymore in order to do its role in terms of regulating gene expression. And this is a great review on temperature sensing in plants. And here you can see phytochrome B that I just mentioned. Basically, its target, one of its targets, is transcription factor P4, which induces the expression of genes that are important for hypocotyl elongation, these extensions that I mentioned in response to shade avoidance, but also warm temperature. So basically what warm temperature does is pushes this back to the basically desensitized form of phi B, so it cannot inhibit, it cannot target P4 for degradation, and therefore it can induce the expression of genes that are important for hypocotyl elongation. And for ELF3 is something similar. High temperature basically leads to ELF3 condensates that can no longer repress P4, so it can basically induce the expression of genes that promote growth. So, and since TZP is part of this complex, potentially it affects these dynamics. So Jack did the original experiments, and then Anna did her whole PhD on this project. And what we saw is that the TZP mutant seems to be hypersensitive to high temperature. So basically, in response to warm temperature, you see an elongation of hypocotyls that I mentioned. And in the TZP mutant, this elongation is more pronounced, especially when you look at the ratio. Whereas when you overexpress TZP, so when you have more TZP than the wild type, the response is hyposensitive. So it's less responsive to this temperature shift from 18 to 32 or 22 to 32. And when we looked at the different wavelengths, so when we replaced white light with blue or red, we could see that the effect was much more pronounced in red light. So in this case, it seems that TZP is actually a negative regulator of thermomorphogenesis and a negative regulator of growth, which is the opposite of what we thought would be happening, because in blue light, we knew TZP is a positive regulator of growth. Anyway, so Tian was able to also rescue the mutant phenotype, so you can see this hypersensitive response basically is gone when you complement the mutant with the fully functional TZP. And Anna also showed that this is not just in terms of hypocotyl elongation. In response to high temperature, there is an acceleration of flowering in wild type. And in the TZP mutant, you can see a hypersensitive response. So it's basically, this flowering acceleration is even, let's say, faster or more pronounced than it is in the wild type. And when you overexpress TZP, so you have more of this protein, you have the opposite effect, again suggesting that TZP is a negative regulator of thermomorphogenesis. And of course, we wanted to see whether TZP operates in one of these two, because these are two different pathways. And in terms of phi B, warm temperature leads to a reduced number of nuclear bodies, whereas ELF3 temperature basically regulates these condensates. So it's the opposite effect that temperature has on phi B localization and ELF3 localization. And we wanted to see how temperature actually affects TZP protein. And we saw that it follows more the pattern of phytochrome B. And it made a lot of sense because we knew that TZP actually interacts directly with phytochrome B. So this is basically counting the number of nuclear bodies per nucleus in response to a temperature shift from 18 to 32 degrees. And here you can see the TZP tagged with GFP, and here it's phytochrome B. And Tian also wanted to see whether temperature affects protein. So in response to a time course at 32 degrees, we could actually see that TZP accumulates. So there's more protein in response to high temperature. So this lack of nuclear bodies is not just because there's less protein. If anything, there is an increase in the total protein levels. And this doesn't happen at the transcript level. And of course, the next question is what happens to the transcription factors that is key in terms of promoting this hypocotyl elongation in response to warm temperature. So P4, that's the direct target of phytochrome B, but also ELF3. So in wild type plants, in col0, in response to high temperature, P4 accumulates, and that's why it induces the expression of all these genes that promote growth and also flowering initiation. Whereas when you overexpress TZP, this is much less pronounced. That again explains the phenotypes that we see. And in the mutant, you can see that even at 18 or 22 degrees, there's almost as if there's some protection of P4. And we had seen similar phenotypes in response to red light, but this is much more pronounced in response to high temperature. And this follows, this explains also the changes we see in gene expression. So these are 3 genes that are important for hypocotyl elongation in response to shade, but also to high temperature. And again, you can see at time course there is an increase of this gene in wild type. And then when you overexpress TZP, this effect is much less pronounced, if any. And then in the mutant, this is much more pronounced. So somehow TZP is affecting P4 levels. We don't know if it's a direct effect. We know that TZP can interact with P4, but it is potentially happening through Phi-B. So we now want to see if TZP can affect Phi-B photochemistry, but also protein levels to see whether it's a direct or indirect effect. And of course, if P4 is stabilized, then you have induction of these genes that I mentioned that promote hypocotyl elongation in response to warm temperature. So at some point, we wanted to see what happens at the whole transcriptome level. So what happens when you have wild type versus TZP mutant and overexpressing TZP lines, and you do RNA-seq when they are growing at 22, or you move them to 32, let's say, for 4 hours. And to our surprise, in addition to these growth-promoting genes that we had suspected that we would see, there was a lot of heat shock proteins. And we still don't use heat stress at this point. It's just 32 degrees. It shouldn't be stressful for the plant, especially if it's exposed only for 4 hours. So this is just to show you the pathway. So this is heat stress. It's from the same review that talks about thermosensing and temperature sensing in plants. And this is a different pathway. It's not thermomorphogenesis anymore, but in response to high temperature, basically you have heat shock factors that then stimulate the expression of heat shock proteins that protect basically the plant from heat stress. And of course, in addition to transcriptional regulation, there's some epigenetic changes. So there's changes at the specific histone variants that have been shown to be another mechanism of thermal regulation. So instead of the canonical H2A, you have H2AZ that is removed, and basically it allows the expression of these heat shock proteins. So what we wanted to see is whether TZP potentially has also a role in thermotolerance. So Michaela started doing these thermotolerance assays. So when you grow plants at 22, they're perfectly happy, and then you treat them for 2 hours, 45, so that's heat stress, and then you basically monitor the recovery. And you can see that indeed, either phenotypically or also if you measure the fresh weight, you can see that TZP mutant basically does better than wild type, whereas the overexpressing line is not doing as well as wild type, and definitely much worse than the mutant. Again, suggesting that somehow TZP promotes thermotolerance, as if maybe it already has some genes that are upregulated at 22 and they're ready to basically promote this thermotolerant effect when the heat stress arrives. And of course, this is something we're investigating further at the moment. We're interested to see if TZP can associate with promoters of these thermotolerance genes. I mean, the genes that we've seen affected by TZP under 22 degrees, they're usually induced. But potentially TZP may have a role as a transcriptional repressor that we haven't identified yet, possibly also through this potential ER motif. And also we want to see if TZP could affect H2AZ occupancy on these thermoregulated genes. We know it associates with chromatin. Plus 3 domains, usually they're involved in chromatin remodeling, so this is something we want to investigate further. And of course, these nuclear bodies, could they potentially be sites, hubs of chromatin regulation? So the idea of transcription factors is quite debatable in the mammalian field, and this is something that I don't think has been investigated much in the plant field. So basically, instead of having proteins moving to the chromatin, to the parts of the DNA that need to be regulated, there is this theory that there's specific loci that are co-regulated, let's say in response to a particular stimulus or by a set of proteins that move to the sides where there is a concentration of these proteins that regulate them. And actually there's some papers showing that there's movements in response to light of loci. So DNA basically moves to the interior or the periphery of the nucleus in order to be activated or repressed, again supporting this hypothesis. And of course, ultimately, although what we work on is fundamental biological research, we don't work on any particular crop species, at least not yet in the lab, because we don't have the expertise or the facilities. We would be interested to see if this particular protein could be a target for improving crop thermotolerance. So TZP is highly conserved in angiosperms, and tomato is a model system that is fantastic for thermomorphogenesis and thermotolerance. So this is something we're looking into through collaborations. So from my talk, some very general concepts. Proteins and pathways can have multiple functions depending on the context. So you can see pathways that are used by multiple environmental stimuli. It's very important to understand the details of molecular processes and in order to appreciate what's the physiological significance of all these different components. And in terms of our research, we want to optimize light capture and plant adaptation without compromising crop yield. Let's say as a bigger picture that, of course, it's something that we cannot do on our own in our lab. And hopefully we can apply some of the knowledge we gain through the proteins that we work on to address some of the key questions in plant biology, to increase crop yield and quality, and also try to improve plant health and environmental resilience. And with that, I would like to help thank my lab and people that did the work that I mentioned today, especially Anna, Tian, and Elisa, and Michaela. They did most of the work that I presented. Our collaborators, so the Herzig lab did all the next generation sequencing. And of course, great mentors. So Professor Joanne Corries, she's been an amazing mentor since I joined her lab and very supportive. Of course, the Gatsby for always encouraging me and making me be part of the family, and of course our funding. And in terms of trying to give you some tips from my experience, and because you're all here today, and I think it's an opportunity you'll be exposed to so many research topics and to so many different practicals. You're going to meet amazing scientists, the plenary sessions, your tutors, the people that organize the practicals, which is a lot of work. So I think whether you stay in academia or not, and whether you're excited by research, I think it's great to find something, a topic that you're passionate about. I think that's the most important thing, because then you will enjoy it. And also, you will hear different career paths. Just keep in mind, every path is unique. You are unique, and you can have a path that suits you at the personal level, your personality, your circumstances, what you want to do in the future, and this might change, at different stages of your career. And also, again, through this network, you can identify great mentors that you can keep in touch with even after the summer school, and also a supportive peer network. Again, exchanging ideas and promoting each other. I think this is something I enjoyed very much, either by being part of the undergraduate, let's say, network when I was at university, again, through the Gatsby, when I was doing my postdoc. I think it's very important to have this mentorship and support. And, you'll always at some point be in a position that there's going to be people that know more than you. There's experts. So don't be afraid to go and ask questions and learn from them. They're here, to help. And then you'll be at a point that you can share your knowledge and teach the next generation on whatever you become an expert on. And again, believe in yourself. If there's something that you want to do, just go for it and never let anyone discourage you. And yeah, it's fun. Yeah, I'll totally recommend plant science and, science in general. So yeah, thank you very much.